Domesticated animals provide key insights into how morphological traits evolve over time. Among them, ornamental and pet animals show how artificial selection dramatically reshape body structures. Recent genomic studies have identified specific genes responsible for these changes. One remarkable example is the twin-tail phenotype in goldfish. This trait was established through artificial selection from wild Crucian carp, Carassius auratus. Our previous research identified the genetic basis of this feature and explained how twin-tail morphology emerged in the context of EvoDevo. I have discussed this in previous video. So in this video, I will briefly review the EvoDevo of twin-tail goldfish. A stop codon mutation in one of two coding paralogous genes is responsible for twin-tail phenotype. This gene duplication event occurred before domestication. The mutated allele in the duplicated coding locus is originally named coding AE127X, but in this video I will refer to it as coding mutant allele. The coding gene is crucial for dorsal ventral patterning in vertebrate species. Without this gene duplication, it may be difficult or even impossible for twin-tail morphology to become genetically fixed. In fact, no stable twin-tail strains have been reported in any other vertebrate species. This brings us to an important question. Why has twin-tail morphology not been found in other fish species? For example, ornamental common carp. Common carp, Cyprinus carpio, have undergone both coding gene duplication and domestication for ornamental purposes. Common carp is the closest relative of goldfish. It possesses coding A and coding B orthologous acquired through allotetraploidization. The ornamental common carp is also one of the most popular ornamental species worldwide. Given its commercial value, breeders would like to have attempted to establish twin-tail variety. Reliable historical records of ornamental common carp breeding exist comparable to those for goldfish. Despite this, no twin-tail varieties of common carp have ever been recorded. While we do not know for certain whether breeders in the past actually attempted to create the twin-tail common carp, it is reasonable to assume that they might have. If you were a breeder, you would like to think the same way. Personally, I would love to see the twin-tail common carp. And if I ever found one for sale, I would definitely want to buy it and try to breed it. But we cannot purchase a twin-tail common carp because no such strain exists. Other ornamental traits such as color, body size, and tameless have independently emerged in different domesticated species. Yet twin-tail morphology have not appeared in common carp. This absence suggests that the mutation does not occur in this species. It raises a key question: why did this mutation arise in goldfish but not in their closest relative, the common carp? Previous studies on zebrafish coding mutant and backcross analysis of twin-tail goldfish have revealed something important. Coding gene mutants show polymorphic phenotypes. In our backcross analysis, some coding A mutant F2 individuals did not display any mutated phenotypes during late development. In genetic terms, this suggests incomplete penetrance. What is the likely reason? The likely reason is the robustness of dorsal ventral patterning. Interestingly, some coding A mutant goldfish embryos initially showed caudal fin mutations. However, these fin folds sometimes reverted to a more wild-type appearance. At the post-embryonic stage, these individuals developed normal-looking caudal fins. This observation suggests something important. The presence or absence of twin-tail morphology might depend on how embryos respond to coding gene deficiency. To test this, we followed a step-by-step approach. First, we generated single and double coding gene deficient goldfish. We knocked down coding A in wild-type goldfish embryos using morpholino injection. Morpholinos are synthetic molecules that bind to specific RNA sequence and prevent translation into protein, and they showed typical coding mutant phenotype. These included weak ventralization and bifurcation fin folds. In some cases, their morphology closely resembles that of twin-tail goldfish at the late embryonic stage. Interestingly, the bifurcated fin fold encoding A morphans later developed into a bifurcated caudal fin with fin rays. This final morphology was nearly identical to that of adult twin-tail goldfish. On the other hand, coding B knockdown in twin-tail goldfish resulted in severe ventralization during embryonic development. This was similar to what has been observed in dino zebrafish mutant. Zebrafish has only a single coding gene, so when this gene is disrupted, most of zebrafish exhibit severe phenotypes and do not survive. The same phenomenon occurred in goldfish when coding B was knocked down. Independent experiments confirmed that injecting 5 to 10 nanogram of coding A morpholino reagent reliably produced these phenotypes. This consistent reproductivity strengthened our conclusion about the role of coding genes in twin-tail formation. Now, let's take a look at the gene expression at the even earlier stage in goldfish. In coding A mutant and morphant goldfish embryos, 3 marker genes showed more ventralized expression compared to controls. This pattern was consistent across different samples. However, things became even more dramatic in coding A and coding B double deficient goldfish. In these embryos, GATA2A and sizzled genes expression expand beyond the ventral half. Meanwhile, FOXB1A expression was limited to very small dorsal region. This strongly suggests that the severe late embryonic phenotypes result from highly ventralized embryonic development. This confirms that our designed morpholino reagent effectively depleted coding gene transcripts in goldfish. In other words, the severe ventralization we observed is directly linked to coding gene loss, reinforcing the crucial role of this gene in dorsal ventral patterning. Now let's examine the findings for the common carp. Since goldfish and common carp are closely related, their coding genes morphants show similar phenotypes. At the late embryonic stage, coding A and coding B single and double morphant common carp exhibited various degree of ventralization. In fact, the coding A and coding B double morphant common carp closely resembled the dino zebrafish mutant. These embryos showed severe ventralization and bifurcated fin folds. Interestingly, these phenotypes also matched the early embryonic gene expression patterns of GATA2A, FOXB1A, and sizzled. This suggests that similar genetic mechanisms are at play in both goldfish and common carp during early development. However, despite their close evolutionary relationship, coding A morphant goldfish and common carp embryos show key differences in the position of the bifurcated fin fold. In goldfish, bifurcated fin fold extends across a wide range of caudal levels. But in common carp, it is restricted to the area around the yolk extension. This difference has important consequences. In goldfish, bifurcated fin fold later develops into a bifurcated caudal fin. But in common carp, this does not happen. Why? The bifurcated fin fold in common carp is positioned too far anteriorly. Because of this, it fails to differentiate into a bifurcated caudal fin during post-embryonic development. We tested this repeatedly using different morpholino reagents, but we never observed a coding amorphant common carp with a phenotype similar to twin-tailed goldfish. In other words, we did not see a bifurcated fin fold at the caudal level without severe ventralization. This suggests something important. In common carp, ventralized gene expression patterns do not directly translate into late or post-embryonic morphology as they do in goldfish. This suggests that coding-related developmental mechanisms are different between these two species, and that difference may explain why twin-tail morphology was a much lower penetrance in common carp. To investigate this further, we compared coding A and coding B gene expression pattern in common carp. In goldfish, these two genes have distinct spatial expression patterns. But in common carp, they are almost overlapping during the early embryonic stages. We confirmed this using probes targeting either the 3' or 5' UTR region. The results were clear. Both probes showed almost identical overlapping expression patterns in early gastrula stage embryos. This points to something significant. Unlike goldfish, coding A and coding B in common carp are not subfunctionalized at the gastrula stage. Subfunctionalization refers to the process in which duplicated genes evolved to take on distinct functions or expression patterns, preventing redundancy. In other words, coding B in common carp can almost entirely compensate for the loss of coding A at this early stage of development. Why does this matter? The gastrula stage and the stages that follows play a crucial role in shaping the caudal embryonic region. This suggests that the differences in coding gene expression between goldfish and common carp are directly responsible for the differences in caudal fin morphology. In goldfish, coding A deficiency leads to twin-tail morphology. But in common carp, coding B compensates for this loss, buffering against the same mutation. This explains why twin-tail morphology easily emerges in goldfish but not in common carp. The recently duplicated coding paralogous genes may have played a key role in the genetic fixation of twin-tail morphology in goldfish. But in common carp, these duplicated genes may have actually prevented the emergence of twin-tail morphology. So, what is causing these differences? It is likely that a combination of cis and trans-regulatory factors as well as differences in coding sequences contribute to how these coding genes are expressed in goldfish and common carp. To pinpoint the key factors behind this divergence, we still have more to investigate. However, based on what we have done so far, we can already draw some interesting conclusions. Even if the coding A gene in common carp had a stop codon mutation, it would likely go unnoticed. Why? Because the overlapping expression of coding B would mask its effects. This means breeders would have no reason to select for it, preventing the mutation from becoming genetically fixed in the population. But what would happen to such a mutated coding A gene? Most likely, it would turn into a pseudogene. A pseudogene is a gene that has lost its function due to mutations. This follows a common trend seen in zebrafish and medaka, where duplicated genes often lose function over time. This raises an intriguing possibility. The presence of two subfunctionalized coding genes in goldfish may have been a key factor enabling the genetic fixation of twin-tail morphology. So, what can we conclude from all of this? Goldfish went through a series of evolutionary events that led to dramatic morphological changes. First, the coding gene duplicated. Then the expression patterns of these genes became subfunctionalized. Finally, breeders selected morphological traits that allowed the coding mutant allele to become genetically fixed in the population. This sequence of events made twin-tail morphology possible. On the other hand, the common carp also underwent coding gene duplication. It was exposed to the same type of selective pressures as goldfish. But one key step was missing, the subfunctionalization of coding gene expression. Without that step, the twin-tail phenotype never emerged in common carp. This highlights how small genetic differences can lead to major morphological outcomes. Now, how do these findings fit into a broader evolutionary context? Comparing our result with data from other vertebrates could reveal how large-scale morphological changes happened in certain lineages. Our previous anatomical studies showed that the bifurcated caudal fin in twin-tail goldfish is supported by a bifurcated axial skeleton. This is important because in all other vertebrate, axial skeletal structures are positioned along the sagittal plane. There are a few other cases where similar reorganization has occurred. For example, paired fins and bilaterally positioned nostrils in jawed vertebrate evolved from structures were originally on the sagittal plane in jawless fish. These kinds of skeletal changes happened early in vertebrate evolution, but they did not happen quickly. Even in domesticated animals with extreme ornamental trait, large-scale skeletal change were rare. For example, genetic studies on dogs and pigeons have shown that the mutations can change their appearance, things like fur patterns, feather shape, and body size. Still, their fundamental skeletal structure remains unchanged. This suggests that even if breeders aimed to develop extreme traits such as twin-tailed dogs or twin-headed pigeons, the genetic framework of these animals might not permit such drastic alterations. Even so, as a matter of fact, throughout evolution, living organisms have undergone major morphological transformations. We observed a striking example of this in the domesticated twin-tailed goldfish. So what could be the key factor behind such changes? One critical factor in this process is gene duplication, which can create opportunity for mutations to influence fundamental body structures. It is widely recognized that gene duplication plays a significant role in increasing phenotypic complexity over long evolutionary timescales. So, gene duplication and genome duplication drive evolution. That's the key, right? Wait, did I actually say that? What we have actually shown is that while gene duplication contributes to goldfish morphological diversification, it has the opposite effect in common carp. We also know that genome duplication occurred in trout and amphibian lineage, yet they do not exhibit extremely unusual morphology. And this might be because they lacked appropriate selective pressures to drive drastic morphological change. So what is the most important implication from our results? Our results imply that the sequential occurrence of evolutionary event in a certain order might enhance drastic large-scale morphological changes. I mean, duplication, subfunctionalization, and subsequent strong selective pressure should happen in a certain order as opposed to occurring as a random isolated events. It is often said that large-scale evolutionary changes take a long time, and this is typically interpreted as a result of the slow accumulation of random mutations. However, based on our finding, I believe that the key factor is not the sheer amount of time required for accumulating random mutations, but rather the rarity of specific mutations occurring the correct sequence. Since opportunity for such rare events take place in extremely limited, large-scale morphological transformation take a long time to emerge. This is the insight I gained from Goldfish EvoDevo about how animal body shape evolve. We tried to create a twin-tail common carp using recent technologies. To be precise, I asked my brilliant colleagues to take on the challenge, yet despite their best efforts, they did not succeed. So this suggests that it's probably quite a difficult task. But in experimental science, proving that something can't be done is much harder than proving it can be done. Maybe someone will suddenly discover a method we haven't tried yet. And who knows, maybe someone out there has already bred this kind of common carp. So if you ever come across a common carp in a pond, take a closer look.